Dr. Powles, preparation slides are ready.
Thank you.
And we move further down now to kidney and bladder.
Thanks for inviting me. I've actually came with a jacket. I looked a lot smarter at the airport. I've been given a really big topic to talk about bladder and renal cancer, which is kind of challenging in half an hour. And so I'm going to get as far as I can and then be dragged off the stage, which wouldn't be the first time in my career. I have a number of disclosures, as you can imagine. I'm going to talk about urothelial cancer first because I guess it's, I don't know, I guess it's more difficult right now. There are some parallels between what I'm going to say and what Kevin said. There are also some distinct differences, and I think there are important differences between these cancers. I'm really nervous about the subset analysis that we're doing in a lot of these cancers. I think we're over-interpreting subset analysis. The reality is, in bladder cancer at least, second-line immune therapy is not that active. It's kind of okay. Remember, chemotherapy wasn't really better than best supportive care. Vinflunine failed to beat best supportive care, second line. And one immune therapy trial has been positive and one immune therapy trial has been negative. So if you're essentially struggling to beat best supportive care, we haven't really hit it out the park yet. And I think sometimes we may get carried away with this. I don't just say this to this audience, I'll say this to any audience, that the bladder cancer story needs a lot of maturation before we've solved that. And I actually think, although bladder cancer is further than head and neck cancer, I'm not convinced that the frontline single agent trials, even in subsets of patients, are going to be positive. I'll explain why later. So this is my first slide, and it kind of just says where we are. So it's a pretty miserable story. You get chemotherapy for a bit, the cancer grows back, you might get maybe a bit more chemotherapy. Only 20% of patients get second-line chemotherapy in bladder cancer and urothelial cancer. People think it's more like 50 or 60. Most patients don't, and that's a subset. This is kind of a complicated slide, but don't focus on it too much. The only interesting thing about it is the color coding. All the drugs you know because you've been described earlier. Two key issues I'd like you to focus on. Number one is we've developed unique biomarker analyses. So when you start showing biomarker data, it becomes incredibly, in fact, it becomes infinitely complex to interpret. I'm not going to give you a 5-minute talk now why it's infinitely complex, but I will just mention a few points. There are distinctions between bladder cancer. Immune cell staining is associated with a good prognosis, tumor staining a poor prognosis. We know that 22C3 and 142 particularly doesn't stain tumor cells. There appear to be technical differences between the Ventana and DACO platforms. The other issues associated with the biomarker is you can set your cut point at different levels. So you can say, I'll define positivity as 10% of immune cells, which is essentially, or Roche are doing 5% immune cells. You can switch across and say durvalumab, 10% immune cells, 10% tumor cells, using a different antibody. And so the options are actually limitless. And my nervousness about this, and I've been involved in this process, is in the phase 2 trials here, we actually went on a fishing trip. We don't talk about this enough. But we went selecting the biomarker that we felt worked best. So the durvalumab program looked at different biomarkers and ended up with a 10% of both using a combined score. And guess what? Their results were astonishingly good. But we know if we perform multiple analyses with biomarkers, we're likely to pick the winners, and that doesn't translate well in randomized phase 3 studies. This is a wedding cake slide. A friend of mine called Charlotte made it in my group, and it's color-coded, which we were impressed with. The outer part of the cake you can see here is the biomarker positive. And the reason I always show it is to show the blue cake, phase 1, phase 2, phase 3. And you can see here as you move across, you can see a lot of variability. You can see in the randomized phase 3, this is the randomized phase 3 pembrolizumab study, the biomarker makes absolutely no difference at all in terms of response. There are some subtle differences in other aspects of the analysis. And that is this randomized phase 3. I'm not going to spend too much detail on this. You can see here that the pembrolizumab is outperforming chemotherapy. These are individuals who have previously failed platinum-based therapy. So this is the second-line setting. This is where chemotherapy fails to beat best supportive care with vinflunine in a randomized phase 3 study. You can see these results, I think, are actually quite good. These are by far the best results we have anywhere in bladder cancer right now. And these results are impressive because the hazard ratio hit 0.73 with patients going into a long-term durable remission. The response rate is nice too. That's higher than you'd expect. But remember, the bar is super low. We then go on to the atezolizumab program. The atezolizumab program pursued a different approach. Kevin talked really eloquently earlier, and he talked nicely about spectacularly failed trials. I have a plethora of spectacularly failed trials. In fact, I've had the highest hazard ratio at ASCO 2 years running. I suspect that may never be beaten. This trial was another trial which I led, which was also negative, but this was more important because unlike the previous studies, which tended to be phase 1 and phase 2s, this was the pivotal practice-changing randomized phase 3. We took a risk in this. We decided to pursue a biomarker-positive approach. People asked me why we did that. And the issue was, with atezolizumab in this setting, we showed in a phase 1 and a phase 2 trial that if you were biomarker positive, you had much higher response rates, double the response rates, and much longer overall survival, double the survival, than you did if you were biomarker negative in single-arm trials. The problem with that is these were not randomized trials, they're single-arm trials. And I told you at the beginning of the talk that the Roche approach, which is pursuing immune cells, we've now discovered that immune staining is associated with good outcomes. And so essentially what we did is we picked the winners. We picked the prognostic winners. We picked the patients who were going to do well whatever happens. And with only 100 patients in each arm, it's inevitable you're going to have a negative randomized phase 3. This is the phase 1 and the phase 2 data showing the biomarker positive patients doing better, and this is a big negative randomized phase 3 trial. You can see from here the response rates are much higher than you'd expect for chemotherapy. Standard response rates for chemotherapy, 5 or 6%. We've got 25%, and you can see here the survival much longer than expected. The ITT population, this is 1,000 patients, the biggest study we ever did. This is exploratory. We talk about it. The EMA and FDA approved the drug based on these data. Whether or not they should have done, discussion for a different day. But the issue is that the blue curve is outperforming the red curve. The quality of life is better. The adverse event profile is better. And in unselected patients, you clearly would want to be in this group than this group. With the knowledge that you already have pembrolizumab with a primary endpoint, a positive randomized phase 3, and a hazard ratio of 0.73, you may choose pembrolizumab in front of atezolizumab, but I would still choose atezolizumab in front of chemotherapy. Remember I said the bar was super low, second-line bladder cancer? The frontline setting is actually a bit more complicated. The bar is a bit higher. I use the word a bit higher because these are 3 groups of patients. These are really good risk patients, intermediate risk patients, and poor risk patients. We stratify patients on whether they're eligible for cisplatin or not. Essentially, we give the losers carboplatin and the winners cisplatin. And guess what? The losers die much more quickly than the winners. That's probably more to do with their prognostic factors than the chemotherapy with drugs we're using, speaking frankly, although some people don't believe that's the case. What I would say is the overall survival of frontline bladder cancer at 12 months is pretty modest, even in the winners. So we do have a lot of work to do here, and there are huge opportunities in frontline bladder cancer. And we know immune therapy is associated with long-term durable remission in subsets of patients. So I would argue, particularly in this group of patients who we know are going to do super badly, it's entirely justified to pursue an aggressive approach looking at frontline immune therapy. The problem with frontline immune therapy is the response rates, unlike chemotherapy, are only about 20%. And actually, the best response, the majority of patients, have progression of disease. So the commonest response is progression of disease. Now, chemotherapy gets in control in probably 60 or 70% of patients. Immune therapy gets in control in 20% of patients. Therefore, the likelihood is, if you go head-to-head against each other, the chemotherapy, particularly at the beginning, is likely to win. We got this curve, and this curve surprised us a lot. This is an overall survival curve. It's only 119 patients, and I'm really nervous about it. But essentially, historical controls, 10-month overall survival. Here we've got 15-month overall survival. Median progression-free survival is only 2 months, and response rate's only 25%. So something must be happening. And as Kevin said earlier, if you give immune therapy first, and then you subsequently give chemotherapy, the chemotherapy works quite nicely. And that's what's happening to this group of patients. These results created a buzz of activity in the academic and the pharmaceutical industry and launched $1 billion worth of studies, 6 randomized phase 3s of 119 patients. We will learn over the next 12 months whether that was a good idea. I think it is a good idea because the questions are valid. The pembrolizumab data is more sobering. Much bigger, 370 studies, 370 patients, less select sites, more patients with comorbidities. And you can see from here the survival only comes down to 11 months. Historical controls, somewhere between 10 and 15. The likelihood is that in unselected patients, this isn't going to work. And we've done 5 randomized phase 3 studies asking that question. Here are 4 of those randomized phase 3s, and you can pick your study. Essentially, 2 main questions are being asked. Each study has single-agent immune therapy, as you would expect, and then each study asks a second question, either CTLA-4 plus PD-L1 or chemotherapy plus PD-L1. Halfway through these trials, the EMA and FDA got in contact with us. And they both said to us, hold on a second, everyone. We're going to change our frontline labels. That 119 patient study and the pembrolizumab study resulted in both atezolizumab and pembrolizumab getting a license in frontline bladder cancer in cisplatin-ineligible patients, in unselected patients. 119 patients did that. The EMA's an odd organisation at times, and I think that was a brave decision because they were very select sites. The randomized phase 3 studies have had a series of interim analyses. It's likely that those studies, the interim analyses of those studies, have prompted the EMA and FDA to make a decision that in biomarker negative patients, PD-L1 negative patients, harm is occurring in the frontline setting compared to chemotherapy. That is the only rationale for the explanation for the changes we've seen in this setting. This is really important because the assumption was only a year ago that everyone was going to be getting frontline immune therapy. And clearly, not only is it no better than chemotherapy, it must be worse. Otherwise, we wouldn't have had to change the trials and also remove the labels at these interim analyses. So I think this hypothesis is fair. Chemotherapy helps most patients a bit. Immune therapy helps a few patients a lot. In the frontline setting, that's a big problem because it means you don't beat chemotherapy. The second-line setting is okay because chemotherapy doesn't win. So clearly, and this is my conclusion, which people are not talking about right now, in the frontline setting, the PD-L1 biomarker works. The EMA and the FDA have both told us that. That means there is a difference between the frontline setting and the second-line setting in bladder cancer. There are lots of hypotheses why that might be the case. The most likely is the tissue becomes invalid due to the period of chemotherapy. And dynamic changes in biomarker expression are occurring with chemotherapy, which means archival tissue in bladder cancer probably is not going to be helpful in the future. This is a slide showing the sequencing of chemotherapy and immune therapy. Focus only on the blue patients. The blue patients get immune therapy and then get chemotherapy. And you can see they respond really well to the chemotherapy. The orange patients have been exposed to chemotherapy and immune therapy previously. When you give them all chemotherapy, it doesn't work. This shows how important it is to sequence these drugs. this is another second message. If you're going to give patients with bladder cancer frontline immune therapy, be absolutely sure they're well enough to sequence the chemotherapy subsequently. Otherwise, they will die more quickly. That's what's happened in these negative randomized phase 3s. That's why the labels have been reduced. This data suggests if you sequence the drugs and you get the chemotherapy in, you can salvage the patients. I suspect what's happened in the randomized phase 3s is we've treated past progression because we believed in pseudo-progression, which doesn't really exist in bladder cancer. And actually, it's not sequencing of immune therapy and chemotherapy in these trials. It's chemotherapy against immune therapy. Many of the immune therapy patients never went on to get chemotherapy, and they die very quickly. This story will tell out over the next 18 months. The complete story will never be told because I suspect we haven't been sequencing these drugs properly because we haven't been broadcasting the lack of cross-resistance between the agents. CTLA-4 PD-L1 in bladder cancer seems to reflect what Kevin said earlier. There is a big question mark. It does not look like renal cancer. It does not look like melanoma. I might be wrong about this, but at the moment, both the durvalumab and tremelimumab and ipilimumab and nivolumab data does not look much better than single-agent therapy. We don't currently have any randomized trials. 3 different approaches to taking this area forward. We can do immune therapy with targeted therapy. This is a study called Biscay. It's not open in Switzerland, but open in different parts of Europe. You do next-generation sequencing, FGF plus durvalumab, PARP plus durvalumab, mTOR plus durvalumab, etc. as you go down. We like this trial. I think it's a good way of cancer discovery, of cancer drug discovery, and we've shown some activity with FGF inhibitors and PARP inhibitors before. As Kevin explained, the chemo combinations are really exciting. We know it's done extremely well in lung cancer. Most people believe the chemotherapy immune combinations will also work in bladder cancer. My feeling is they are likely to work too, particularly for overall survival. I'm drawing my bladder cancer story to an end to get 7 minutes of renal cancer in. Just going to talk a little bit about resistance. We've talked a lot about activity in the past. So we can show DNA replication, cell cycle genes, T-cell effector genes are often associated with high response. We've spent a lot of time showing TGF-beta is associated with resistance. Bladder cancer you can split into 3 immune phenotypes. You can see here an excluded group, an infiltrated group, and an immune desert. And in this excluded group that's common in bladder cancer, you can see, unlike the other 2 groups, TGF-beta seems to be important in generating immune infiltration. We went on and did animal model experiments to show that was the case. So we now show that T effector signatures and tumor mutational burden, both important with response, TGF-beta important in resistance. We've actually done a little bit of neoadjuvant work in this setting too. So this is a neoadjuvant study, 6 weeks of atezolizumab prior to surgery. These patients are not eligible for cisplatin-based therapy. This neoadjuvant approach hasn't really been talked about much. There's a bit of lung cancer data. We kind of like the approach, and we've shown very high response path CR rates, so 30% path CR rates, 40% path CR rates in biomarker-positive populations. I think the future is going to be around perioperative immune therapy, and I can see us in bladder cancer giving a couple of cycles of immune therapy prior to cystectomy, and then giving more immune therapy plus or minus chemotherapy afterwards. We're showing some really interesting things in the tumor bed, so you can see this intense immune infiltrate, which wasn't there before, and we are now defining new parameters of major pathological response for these patients. We're also showing dynamic changes to PD-L1 and CD8 cells. So that dynamic biomarker issue I talked around with chemotherapy, we're showing that after only 2 cycles of atezolizumab. So in in bladder cancer, immune therapy is standard for platinum refractory disease. The biomarker doesn't work. In the frontline setting, it's going to be very difficult. We're going to have to use combinations, but the biomarker does work, and then treatment in earlier disease is needed. I've only got about 4 minutes now. I'm actually probably just going to talk about this, about CTLA-4 PD-L1, because it's really important, and the EMA's made a really unusual decision. The CTLA-4 PD-L1 story is exciting. I've said in bladder cancer and in head and neck cancer it doesn't work. In kidney cancer, the phase 2 data was extraordinary, was really good. And so we did a randomized phase 3 trial of ipi-nivo versus sunitinib. This is a frontline study for renal cancer. There can't be a more important trial. It focuses on intermediate and poor risk patients. And you can see from this hazard ratio of survival of 0.63, which is essentially a 37% improvement in survival compared to the previous standard of care. You can see a higher response rate and a trend towards better progression-free survival. Beating sunitinib, which is a progression-free survival drug, was always going to be really hard. These results are extremely good, by far the best renal cancer results we've ever had while I've been treating kidney cancer. The biomarker seems to work here as well. Hazard ratio is going down to 0.45. Kevin earlier talked about odd decisions by the EMA. You know, this would be a population which clearly patients would want. It wasn't the primary endpoint. But when you're getting hazard ratios below 0.5 for a common cancer, you need to be thinking very carefully about why you're not giving these patients treatment. The toxicity profile, as I said before, grade 3 or 4, adverse events were lower. The quality of life was better. This was rejected by the EMA. I don't know what to say. So I've been involved in this process, and there is information in the public domain. The reason the EMA has rejected this is because there was no third arm, there was no nivolumab arm, and there was no proof that nivolumab alone wouldn't have beaten sunitinib. That was the rationale. There is now a debate going on in the clinical community about this result, and I'm interested to hear what you think in Switzerland. I know you've approved it in Switzerland, it's approved in the United States of America. This is how the debate goes. The letter of the law is the EMA has rules, and they have not abided by the rules. The rules are that you need to use monotherapy. You need to look at the component parts before you move on to test combinations to ensure that you're not putting patients in harm's way or giving drugs unnecessarily for a toxicity or from a cost perspective. The other part of the argument is that a combination, irrespective of the detail, that has a survival hazard ratio of 0.63 or 0.41, depending on the population, which is better tolerated with better quality of life, from a patient's perspective, it doesn't really matter. And people have made analogies. One of those analogies would be an aeroplane. It would be inexplicable that an aeroplane authority, given the choice between two aeroplanes, one which had an increased chance of death compared to the other, would approve the aeroplane with an increased chance of death. I wouldn't get on that plane on the way home if I knew it was less safe than another plane, irrespective of how it was made, number one. The other argument that's being made, the other analogy, is the boat and the life raft. So you've got a sinking boat, the boat is sinking. You have life jackets and you have life rafts. And the EMA rejects both of those because we haven't told which of the two are important. But as we've shown, both tend to be relevant. So those are the arguments which are currently being said in this setting. I'm going to stop talking now because I want to have 5 minutes of questions before your break. I'm very keen to take any questions on VEGF-targeted therapy and immune therapy, which I've not talked about. I'm really interested to hear what you think about this EMA decision. I know where I stand because I'm kind of patient-focused, but I can see the other side of the argument as well. And clearly, I'm very happy to talk about some of the very controversial things I've said today in bladder cancer, some of which may pan out and some of which may not. Many thanks for your attention.
I like the boat one. So, any comments from your side on this decision, Solange?
It's shocking. I was contacted by your colleagues in terms of trying to intervene in the name of big organizations like ESMO for this. Like you described it very well, it's an undue decision. I mean, it's about patients' survival, quality of life, and everything you can evoke. And when you have a very well-conducted trial, it would be a magnitude of benefit of 5. It would be an evidence 1A without any doubt. You are not supposed to insert in your trial design a potential new hypothesis when you are a regulatory authority. When you are an investigator, it's something else, right? So if you start to accept this movement by a regulatory authority, they can invent whatever they want, because there's always still another plan which can be evoked. Problem of ESMO was more to say that it's politically difficult for such an organization to enter this political discussion with the regulatory bodies. But mounting or helping the community and the patients' advocacy group also to try to fight against this decision is really priority. 2 days before, I went to EMA for another trial, which was also positive, where the EMA subjectively decided to look at unplanned subgroup analysis and to exclude the subgroup, which was unplanned post-hoc, right? Same thing. I mean, some patients might have benefited from the strategy and will not have access. That's a general move of EMA now. With immunotherapy, they try to reduce the indication as much as possible, probably for cost-effectiveness story. We don't have these trials, and in general for costs. So that's really something we will have to deal with. EMA is 6 months delayed or 18 months delayed in between as compared to FDA, and tends now to reject some evidence-based data, whatever the magnitude and whatever's conduction, how the quality of the trial. So it's a new era for us, right? We'll have to fight against something which is really kind of an unfair judgment about the clinical trial. And that's something politically interesting.
I have a question. You left out VEGF targeting, and I'd be very interested to get your opinion on that. You know, I presented the quadruple treatment in advanced non-small cell lung cancer. We worked on bevacizumab together with targeted treatment. I'd be very interested in your thought about using it as combination in renal cell cancer with immunotherapy.
So is anyone showing that slide? So this is the axitinib pembrolizumab data, and I think that the VEGF story goes like this. The story started with the investigation of bevacizumab, which is a monoclonal antibody to the ligand, in combination with atezolizumab. The results with that combination showed modest response rates in the region of 35–40% compared to sunitinib, maybe a little bit higher. They showed modest progression-free survival in unselected patients. They did a randomized phase 2, and that randomized phase 2 showed that in the biomarker-positive population, you could outperform sunitinib with bevacizumab and atezolizumab. And so Roche performed a randomized phase 3 study in that biomarker-positive population, which is now in the public domain, and the PD-L1 positive PFS is indeed positive. They've done a randomized phase 2 with a monotherapy, so they'll probably get through this hurdle showing that, because the monotherapy didn't work well. The BMS people with ipi-nivo have come back and said, well, look, atezolizumab as a monotherapy didn't work, so why would a nivolumab work? That's been one of their arguments for not doing the third arm in the trial. Nevertheless, Bev-Atezo looks from a PFS perspective to be modestly better in selected patients. Remember, that's bevacizumab, and then, oh, here we go. And then when you look at axitinib and pembrolizumab, you're seeing these astonishing results. So instead of response rates in the 30 and 40%, you're seeing response rates of 70%. This is a VEGF TKI rather than a monoclonal antibody, but the two drugs are behaving very differently. It looks like bevacizumab genuinely does have an immune component to it. There was a publication in Nature Medicine. I haven't got slides on that today. But that Nature Medicine's paper showed, compared to sunitinib, there's a T effector signature associated with bevacizumab activity. And axitinib is clearly a much dirtier drug, probably a more powerful VEGF-targeted drug. And the question here is whether or not, these results look different. The likelihood is that axitinib and pembrolizumab is going to be positive for response, progression-free, and overall survival in unselected patients. And it's beginning to draw the hypothesis that bevacizumab may be working more as an immune therapy rather than a VEGF-targeted therapy in this environment. We've got a bit of supporting data for that. We've got a few bits and pieces coming out in the not-too-distant future about subsequent therapy after Bev-Atezo, and response rates for sunitinib and axitinib are very high. Whereas if you re-challenge after Axi-Pembro with sunitinib and pazopanib, response rates are actually pretty poor. So we're beginning to show differentials between VEGF-targeted therapies. There are a lot of these trials. There's Axi-Pembro, there's nivolumab, cabozantinib, there's lenvatinib, pembrolizumab, there's evalulimab, axitinib. Again, just like the bladder program, these sorts of data have sailed 1,000 ships. But I think these trials are very likely to be positive. I've got some underlying reservations about evalulimab and its ADCC activity, and therefore I'm not convinced it's going to be as active as pembrolizumab. I don't know that yet. I hope it is. It's not a Pfizer thing. I lead one of their trials, and I'm very excited about that particular study. I'm not sure all drugs are the same, but the bottom line is I think the evalulimab axitinib study will be the next, will probably be Pfizer's first big successful randomized phase 3, and I think evalulimab will end up getting a license in this setting.
Thank you. I think it's time for